Hello and welcome. On behalf of CME Outfitters, I'd like to welcome and thank you for joining us for episode 2 of the CMEOCast series titled Safety and Efficacy of Emerging Treatments for the Management of SLE. I'm Alan Gabalski, Professor of Medicine at Weill Cornell Medicine, an attending rheumatologist and co-director of the Clinic for Inflammatory Arthritis at Hospital for Special Surgery in New York. I'm joined today by my friend and colleague, Dr. Richard Fury. Dr. Fury is Professor of Medicine at the Zucker School of Medicine at Hofstra Northwell and Chief of the Division of Rheumatology at Northwell Health in Great Neck, New York. Welcome, Rich.
Thank you, Alan.
In addition to CME and CE credit, today's activity is also eligible for ABIM maintenance of certification points. Once you complete the activity, provide your ABIM ID and birth date when completing the evaluation. CME Outfitters will submit your mock points. You can also claim this activity as a CME for MIPS improvement activities. Over the next 90 days, actively work to incorporate the improvements in your practice presented in today's activity. CME Outfitters will send you a follow-up survey in approximately 3 months, and upon completion of the survey, CME Outfitters will send your confirmation to submit to CMS, attesting to your completion of a CME activity for MIPS improvement certification. Now, in episode 1, we discussed the immune pathways that helped us better understand lupus, as well as informed targets for drug development. In this episode, we'll evaluate the safety and efficacy of emerging therapies for the management of SLE. Rich, let's begin with a review of the EULAR and ACR classification criteria for lupus.
We've had several iterations of classification criteria, and the EULAR-ACR classification criteria are the latest. Now, it's important to point out diagnosis versus classification. Diagnosis of lupus is in the eye of the beholder, whereas classification criteria are important for standardizing entry criteria into cohorts and into studies. But we'll use classification criteria often to guide us in making a diagnosis. And as mentioned, these are the latest classification criteria. There are certain rules. One cannot be classified as having lupus unless they've had an ANA of 1 to 80 or greater at some point in their course of their disease. And as you can see in that middle row, if absent, do not classify as lupus. And a lot of people might object to that in diagnosing, but that's where diagnosis versus classification comes in. And then you see some additional rules down in the lower box. But it will make more sense if we take a look at some of the items. And basically, in order to be classified as lupus, one has to have 10 points or greater. And you, in each of these categories, in each of these boxes, you take the highest scoring item and add them up. And there has to be at least one clinical item in making the classification, in meeting the classification criteria. So just when you memorize the last classification criteria, there's now a new set. All right, what I'd like to talk about now are outcome measures in lupus, specifically disease activity instruments. And I'll be very honest with you, this can be a little bit dry, but it's so important for you to understand these outcome measures in interpreting clinical trials. And there's unprecedented clinical trial activity these days, which means a lot of publications. And if you're going to understand the outcomes, you have to understand these disease activity instruments. So there are different measures that we use. One measures disease activity. We incorporate health-related quality of life measurements into our clinical trials. And not that we see damage accrual in, say, a one-year study, but in some of the long-term extensions that go multiple years, we do see damage accrual. And damage is defined as an irreversible change in the organ system that occurred since the onset of lupus. Now, I'll be the first to admit, when I see patients, and I see a lot of lupus patients, I don't use disease activity measurements. I do it sort of informally and might categorize them as mild, moderate, severe. I'm not even sure I do that. But there are formal disease activity instruments. The two that have risen to the top are the SLE and the BILAG. The SLE originating in Toronto and the BILAG in the British Isles. And then we've made life even more complicated by incorporating these disease activity instruments into composite indices. And those composite indices are known as the SRI, which stands for the SLE Responder Index, and the BICLA, standing for the BILAG-based Composite Lupus Assessment. All right, the SLEDAI evaluates 24 lupus manifestations, and a major rule is that you don't score it, that particular item, unless it's attributed to active lupus. And the items are weighted with scores ranging from 1, for example, thrombocytopenia gets a 1 point. Arthritis gets 4 points. Anyway, you add all the scores up and you have a total. And as a rough guide, 13 to 20 is severe, 6 to 12 points is moderate. Now, if we're talking about improvement, one can only lose points if there's complete elimination of disease activity in that particular, for that particular item, or resolution of a laboratory abnormality. So for example, if someone has a head to toe wicked discoid rash and it gets 95% better, I mean, that would be terrific, except you'd still score the same 2 points for rash because there's still some residual rash. So it does require complete elimination. Now, what's a clinically meaningful improvement in the total SLEDAI score? Well, you'll get different answers from different investigators, but it ranges somewhere between 3 and 7 points. Now, the other measure, BILAG, is a little bit more complicated. It measures improvement and worsening by organ system, by different domains, and there are 9 domains. And in total, there are about 100 parameters. And you score compared to the prior visit, say a month before, as not present, improving, same, worse, or new. It does take a little bit more time than doing the SLEDAI. And then each domain, each of the 9 domains is scored a letter score based on a detailed set of rules. And I have to keep my BILAG Bible next to me when I try to score these domains. But as a rough idea, an A score represents severe disease activity, and then an E score, no activity. As I said, we made life a little bit more complicated. This was done as part of the belimumab program many years ago. We had a composite index, the SRI, SLE Responder Index, as the primary endpoint in phase 3. And composite indices were being used in osteoarthritis and Crohn's disease and rheumatoid arthritis, so why not lupus? And it certainly makes sense. In order to be considered a responder, and it was dichotomous, so either you responded or you did not respond. One had to have a 4-point or greater improvement in the SLEDAI score. There could not be any worsening, and that's important. So you couldn't be successful with one item, say rash going away, and then develop renal failure. That would not be a success. So there could not be BILAG worsening in other domains. And then overall, there couldn't be worsening in the physician global assessment. There was a little bit of wiggle room, about 10%, which calculated to 0.3 points. All right, and then there was a similar composite, but using the BILAG as the driver of efficacy instead of the SLEDAI. And that's known as the BICLA. And I'm not going to take time to go through all of these items, but it was the same idea. There had to be improvement. There couldn't be worsening. And this was developed during the epertuzumab program. And although the epertuzumab program, an antibody to CD22 on B cells, was a failure, the BICLA is certainly alive these days and has been the primary endpoint for some of our recent studies. Now, there are many other instruments that we use in clinical trials. They're all listed here. We have skin-specific, such as the CLASI. We have a flare index. We use the patient global assessment, quality of life measurements, such as the SF-36. We measure fatigue, and we measure damage.
So Rich, before we go into a discussion of the novel agents that are coming into our therapeutics, review for us the current landscape of agents we have available.
Well, our toolbox is somewhat restricted. And you can see on this slide that actually the top 3 bullets, these drugs were grandfathered in 1952 by the FDA. Then along came some of our immunosuppressives, and they certainly have made a big difference. And also cyclophosphamide for our sickest patients. It was about 10 years ago that belimumab got an approval. And I have rituximab on this list. And though it's not FDA approved for lupus, it sees some use for certain situations. There are other drugs, thalidomide, lenalidomide, quinacrine, an antimalarial that sometimes see use. And then we have adjunctive therapies, such as ACE inhibitors for our patients with proteinuria, bisphosphonates. I mean, osteoporosis is a big issue in lupus, either because of the disease itself or because of the steroids that we use to treat the disease.
So with this in mind, Rich, what then are the greatest unmet needs for our patients with lupus?
Well, we still have a ways to go. If you go back to the late 1940s, we didn't really have therapies for our patients with lupus, and mortality was about 50% at 7 years. I wasn't around back then, but I imagine most patients who died because of lupus kidney disease. And though we've made significant inroads, we're still losing close to 20% of our patients 15 years into their disease. And if you look at comparisons between mortality rates in our lupus patients versus age-standardized mortality rates, you see a decrease in the lupus patients, but it's not as great as the non-lupus patients. In fact, of those aged 15 to 24 years, lupus as a cause of death ranks 10th, 5th amongst Black and Hispanic females, and it actually ranks 1st amongst chronic inflammatory diseases. And then turning back to lupus nephritis, we're only seeing about 1/3 of our patients achieve a complete renal response at 1 year with our state-of-the-art therapies. And in fact, if you focus on those with proliferative disease, specifically class 4 lupus nephritis, almost 44% are developing end-stage kidney disease at 15 years. So we do have major unmet needs. I think the biggest need is actually in lupus nephritis, followed by moderate to severe extrarenal disease. On the wish list, we need to prevent damage, and that damage could either be from the activity of the underlying disease, or it could be from the drugs that we use to treat the disease, specifically steroids. And we'd love, and we're actually talking about remission induction. So we'd love to see remission.
So Rich, that brings us to the center of this presentation, which is the emerging treatments to try and meet your request for agents to fill unmet needs. Why don't you walk us through the data for some of the newer drugs that we've been seeing at our meetings?
Yeah, the way I've divided this section is into SLE versus lupus nephritis. So we'll talk about SLE first. In front of the FDA, and maybe we'll hear something around mid-2021, is the fate of anifrolumab. If we go back about 4 or 5 years ago, I presented the phase 2 anifrolumab data at the American College of Rheumatology meeting, and it was very exciting. All right, so first off, what is anifrolumab? It's an antibody to the type 1 interferon receptor. So it blocks all 5 subtypes of type 1 interferons. And we know that interferons play a major role, as I showed you in my cartoon, in the pathogenesis of lupus. So based upon the very exciting phase 2 data, the phase 3 program was undertaken, and the studies were known as TULIP-1 and TULIP-2. They were pretty much identically designed. The exception was TULIP-1 studied 2 different doses, the 300 milligram dose, and then a lower dose at 150 milligrams, and the third arm was placebo. TULIP-2 studied just 2 doses, the 300 milligram dose, and then placebo. And when I say placebo, you have to remember that these patients come in on standard of care. So they're getting their background steroids, antimalarials, immunosuppressants. We do not take those away. So it's really not just placebo, it's standard of care arm. All right, but back to the studies. TULIP-1 had as its primary endpoint the SRI. And the reason for the SRI was that the phase 2 study had as its endpoint the SRI, and the data were terrific. So why interfere with something that's working? However, TULIP-1 results came in quicker than TULIP-2 because the recruitment went quicker with TULIP-1. And TULIP-1 did not reach its primary endpoint of SRI. But what we discovered is that the other composite index, the BICLA, worked just fine. So the question was what to do with TULIP-2. Do we change the endpoint prior to unblinding of the data, or take our chances and leave it as SRI? Well, what we decided to do was to change the primary endpoint to BICLA, and I'll show you the data. And I should emphasize once again, the change in the endpoint was before unblinding. Here are the data. I don't think we have time to go through all of these numbers, but the primary endpoint, as I just mentioned, was the BICLA response at week 52. And if you look at the far right, the last two columns tell it all. It has the difference, the effect size, and then the p-value in the far right. So a very successful study. And then you see secondary endpoints. The BICLA response and the interferon high at baseline, steroid reduction, skin activity, swollen and tender joints. And the top three of the secondary endpoints were successful. Joints did not work in this study, however, joints worked in TULIP-1. And that may just speak to our evaluation of arthritis and lupus, which is very difficult. And here are the adverse events, pretty well tolerated drug. The herpes zoster stood out. Shingles was more common. We actually saw that in phase 2, and that was repeated in phase 3. In the treatment arm, 7% frequency of shingles versus 1% in the placebo arm. And there was a higher frequency of respiratory infections. But all of these shingles was treatable with medication. So we put everything together, TULIP-1, TULIP-2, and the phase 2 study known as MUSE, with the two composite indices, SRI and BICLA. 5 out of 6 were successful. The exception was the primary endpoint for TULIP-1. So, I mean, I do see these as positive data for sure. And having done lupus trials for 25 years now, lupus trials are really tough. And essentially all trials, with the exception of the belimumab trials, have failed. So here we see positive data. And I do think overall, if one looks at the totality of the data, anifrolumab is a success.
So Rich, at the top of your list of unmet needs was lupus nephritis. We have data from 3 promising trials, and we're excited about the very recent approval of voclosporin for lupus nephritis. Let's start with a new kid on the block and the Aurora trial.
So voclosporin is a calcineurin inhibitor. And talking about miracles, here we see another miracle. The Aurora study had a positive primary endpoint response, and all the secondary endpoints were successful as well.
Rich, you were the lead author on the BLISS lupus nephritis and nobility trials in lupus nephritis. Can you talk a little bit about those?
There were signals from the phase 3 program with belimumab in SLE that there were some favorable effects on kidney disease. So a kidney-specific study was developed, and BLISS-LN was the largest lupus nephritis trial ever done. The primary endpoint was something called PER, primary endpoint of renal response, and it had a 2-year endpoint. And you'll see if you focus on the middle column, the treatment difference, the effect size is listed for you, just shy of 11 percentage points. And the p-values on the far right. The primary hit, and the primary was based upon a reduction in proteinuria to a protein creatinine ratio of 0.7 or less, There were some other requirements to meet that endpoint that were actually very rigorous. Then you see the secondary endpoints, complete renal response at 2 years, the primary endpoint of renal response at 1 year. So both of those were successful. And then an interesting endpoint was the time to renal related event or death, basically a surrogate for worsening or flare. And that's expressed as a hazard ratio, which you see in the next to last column. And there was a 50% reduction in this particular item. So primary hit and all the secondaries hit. Pretty much a miracle for lupus. And then lastly, these are phase 2 data with obinutuzumab. And obinutuzumab is a third generation B cell depleting agent. So I kind of refer to it as granddaughter or grandson of rituximab. But it does deplete B cells far more potently than rituximab. And this has 2-year data on the slide. If you look at 1 year, and in this particular study, obinutuzumab was given on a background of mycophenolate, and the comparison was placebo and mycophenolate. We see an effect size of about 12 percentage points at 1 year, which grew to 22 percentage points at 1.5 years. And then at 2 years, we see a very similar effect size as we did at week 76. But the interesting fact is that the patients were treated at baseline. They were treated just like in the rituximab regimen, 2 doses, 2 weeks apart. They received their second course of therapy at 6 months. So still 18 months after their second course, we see an effect size of 20 percentage points or close to it. The B cell depletion data tells us that obinutuzumab is far more potent than rituximab, and that could be why we see better responses here than we did in the LUNAR trial with rituximab.
Any other agents that we should keep on our radar screen that are up and coming or recruiting in clinical trials?
Well, there's amazing amount of activity right now in lupus and lupus nephritis, and I'd love to list every single one, and I have it on this slide, at least some of them. So I've divided into lupus versus lupus nephritis. What amazes me right now is how eclectic the attack is on lupus. And I showed you in my cartoon all the different potential targets, and that was only, half the game. There are many more targets. So just to run through some of these agents, in SLE, there's combination therapy, rituximab and belimumab. Baricitinib, we should see some data perhaps in the next year. In lupus, dapirolizumab, an antibody to CD40 ligand, is entering phase 3. And iberdomide, which is yet another strategy for attacking lupus, should be entering phase 3 because of recently successful phase 2 data. Turning to lupus nephritis, we talked a lot about anifrolumab. Well, anifrolumab has been studied in lupus nephritis, and those data should be forthcoming in the next half year. Antibody to CD40 is under investigation. Those data as well in lupus nephritis should be forthcoming over the next half year. And a real mouthful, deucravacitinib, which is a TYK2 inhibitor, is also being studied in lupus nephritis. And there are other drugs being studied in lupus nephritis that aren't as far along as the ones I've shown you. But it's really exciting times in lupus for sure.
Well, it certainly is, Rich. What are the takeaways from this that you'd like to leave the audience with?
Well, we've done better than in 1950. There's no question there's been a reduction in mortality, but we kind of hit a plateau. We're still losing patients. Maybe at 15 to 20 years, there's still a mortality rate of about 18%. So that's a major unmet need. And we're losing patients because of infections from some of the side effects of these drugs. We're losing patients because of increased cardiovascular disease. But I would still say the major unmet need is in lupus nephritis and moderate to severe lupus. And as I've said repeatedly through this presentation, The drug development activities right now have never been seen like we are seeing now. And so I think what this translates into is a much brighter future for our patients with lupus. There's no question that outcomes will be better as more and more therapies become available to us.
Well, thank you again, Rich. This has been a great presentation. And audience, please join us for episode 3, where we will be discussing principles of shared decision-making for patients with lupus. All 3 episodes and a wide variety of education and resources on inflammatory disorders are available on the CME Outfitters Rheumatology Education Hub for both healthcare providers and patients. Now again, to collect your credit for today's activity, you can click on the link to complete the evaluation, and you'll then be able to print your certificate from the platform. And as a reminder, if you'd like to apply for ABIM maintenance or certification credit, provide your ABIM ID and birth date when completing the evaluation, and CME Outfitters will submit your maintenance or certification points. Thank you for joining us today.